Been told that I can carry on till 6 maybe, but I understand several of us are teachers. You may not be probably be able to digest content till 6. So we'll see, and a short break in between maybe. So by the time we end up today's talk or today's sharing of information, you will probably be in a situation to maybe look into the organism that you are trying to work on with more intent, with more interest maybe, right? So this is the whole idea, and there is some information which you might also already be aware of, but then as I said, the whole idea is to encourage you to look into your animal or your plant species from a different perspective, and the perspective of model organism for that matter. So thank you very much for the kind introduction. Let us start with the discussion. So my first question, if you are allowed to write something in the chat boxes, and if you could, then I would be happy to see that also. Can a few of you write down maybe the animal or plant species you have decided to work on? I'm sure by the time when you are attending this coursework, you already have decided something. I mean, the animal or the plant species that you would be working on. So I have just opened, yes, I have just opened the chat box and I can see Rajendra typing beetles. So he has decided to work on beetles. There are a few more participants here. They would be working on something else. And if Rajendra says beetle, has he identified any particular genus or species of that genus for that matter? Uh, Furzan Yazdi has identified Katla to be very specific. Poonam Lonkar has identified Drosophila. That's great. Then cowpea, nematodes. Great, great, great. Okay. Buffonidae, the family. Okay. Then Rhodania royale genus. Okay. Swapnil Patil. And okay. So we can see from insects to fish to beetles to arthropods to you know vertebrates. Yeah, zebrafish. Good. Cystodes. Yes, very nice. Fine. Good. Good. Okay. Wanted few more. Wanted few more. Arabidopsis. Fine. I'm not asking the name of model organisms. I'm asking what have you decided to work on, Shruti? Probably you have mistaken my question. Okay, anyways, let us go further. I'm not at all asking you what are all the model organisms. I'm asking you what have you decided to work on? What are the animal or plant species you will be working on in your doctoral work? Anyways, so I understand that there is a good variety. Now, when you will defend your viva or when you will present your, yeah, somebody, Wagmude sir, he's planning to work on sheep, maybe a variety of sheep. Yes. So when you'll be defending your doctoral work, microcrustaceans are there, Shruti. Right. Now you're to the point, Shruti. Good. So as I said, when you will be defending your work, and even when you will be working on the animal or plant species which you have decided to, the question that will be asked and should crop up every time when you are working on the animal or plant species of your choice is, why did you choose that particular organism for your research? There has to be a reason, right? Now there can be several reasons. There can be reason because probably like somebody is working on katla. The reason simply can be because katla is a economically important fish. Yeah, it is directly associated with nutrition. Somebody can probably be interested in the biodiversity aspect. Somebody like Karishma probably would be interested in parasitological aspect of the animal organism she is working on, right? So there can be several reasons for which you have chosen your organism. But if you look into the history of the findings of biological research, you would see that there are some favorites. There are some favorites in the sense that if say for example I talk about fertilization in animals, yeah, fertilization of sperm and ovum, immediately what comes to your mind is, in the chat box please, Lilly, the work of Lilly, fertilization of sperm and ovum, fertilization theory of Lilly. What comes to your mind? Which organism comes to your mind? Any answer? Okay, I'm still waiting. If not, then I'll tell you, because sometimes there is a lag. So Pawan is planning to work on honeybee. Probably he's planning to working on honeybee. The answer is not honeybee to what fertilization I was talking about. The answer to my question is sea urchin, right? So most of the work that was carried out with regards to fertilization in animals was carried out on sea urchin, especially by Lilly, right? So similarly, if we talk about, say, genetics, sex-linked inheritance, what comes to your mind? Ah, fantastic, Vaishnavi. Yes, Drosophila. Right. So when you're talking about sex-linked inheritance, genetics, what comes to your mind is a Drosophila, a fly, fruit fly. Yes, Manisha, madam, right. So then you associate a particular kind of work, a particular kind of research. Similarly, because there are students from botany also, you may talk about jumping genes, transposons. What comes to your mind? Or cytoplasmic inheritance, if there are any botanists over here. Yeah, fantastic, Vaishnavi, right? Again, maze, Z maze, right? So that is what I was trying to tell you. So if you associate a particular kind of work that has been carried out already, you would see that there are some favorites. And these favorites are called as model organisms. So in crude terms, I'm calling these as favorites. Now, what makes them favorite? Why a particular organism becomes so much favorite, right? Let me see if my slide can change. I do not know why my slide is not changing. Give me a moment. Unmute yourself and let me know if the slide has changed at your end or not. Can you see a new slide, model organism?
Yes, sir. Yes, sir.
Now I'll be unable to see your chat boxes, unfortunately, but you can unmute and discuss with me or answer the questions also. So yes, the concept of model organism. Now, I usually, when I make presentations, I choose images very carefully. And I was looking for a mouse, and I found this image. Of course, one which does not compromises the copyright, so I can use this as an unlicensed one. And I liked this image for a reason. When we are looking at an organism from the aspect of a model organism, we are interested in how the findings of this organism can be correlated with the systems in a human being. So here I see this mouse reading a newspaper, something like we do in the morning. I mean, this generation does not read newspapers, but we do, and we used to. So this is something like human, right? So that also goes with model organisms. So if you have a finding in an organism, say for example a Drosophila, or maybe C. elegans, or maybe Arabidopsis, like somebody was saying, can you see these findings in other organisms? And because we are human and we are very selfish, we want to see the replicas of those findings in human beings as well. So point number one is definitely that these model organisms are non-humans. Then that means when we are choosing a model organism, we are not talking about human beings, we are talking about some non-human organisms. And why do we do so? We are studying whatever names you are talking about. So, like I said, Drosophila or Caenorhabditis elegans or any other model organism, we study them to understand several biological processes. Now, Through the course of this presentation that I am delivering, you would see that there's not one biological process as such, but there are several biological process, including the physiologicals, metabolic process, development process, which can be studied in a lot of depth and can be correlated with what is happening in human beings, right? So like I said, the findings you would have in the model organisms will give you more insights into working on the same aspects in other organisms as well. So if you have probably made one finding in sea urchin with respect to the fertilization, probably you would look into the fertilization in human beings from that perspective as to what model is implemented in sea urchin and what can happen in human beings. Same goes with, say, for example, cell cycle. We all know that Saccharomyces cerevisiae was one of those fungal species, yeast species, which was studied with respect to the cell cycle, as we'll be discussing further, with those checkpoints in the cell cycle. And the same were found in human beings as well, right? And these model organisms, they occupy a central position, an important position in phylogeny as well. When we say phylogeny, we are talking about evolutionary tree. So they are linked somewhere. They establish important links, say, for example, right from invertebrates to vertebrates in case of animal kingdom, right? So this is how the importance of model organism can be ascertained. But how do we establish that an organism can function as a model organism? What makes an organism a model organism, right? So in the image which has been displayed on the right hand side of your screen, you can see several of them. There is one plant species which is called as mustard plant, Arabidopsis thaliana. Then there are several invertebrates, the roundworm Caenorhabditis elegans, a very famous model organism. Then there is an arthropod dipteran insect fruit fly, which is scientifically called as Drosophila melanogaster. Then there is a different kingdom altogether, east, Saccharomyces cerevisiae. Then there is a vertebrate commonly called as zebra fish because of the bands which it has, Denio. And then there is a mammal, mus musculus, that is the mouse, and a frog, another vertebrate, which is an amphibian, Xenopus laevis. So these are some of the model organisms which have been displayed in the adjoining image, and all of them have been shown to correlate at the molecular level. See that DNA signifies the molecular level, at the molecular level with the human being. So we are interested in the findings of these organisms and associate these findings at molecular level with the human being. Now, as I said, what makes an organism a model organism? I'll go very gradually and slowly so that you understand the whole concept. I understand there will be an examination as well for your coursework. So you should be able to answer these small petty questions, but important questions, right? So one aspect that makes organism a model organism is a size, because you see, can you see an elephant as a model animal? Now just think over it. Can you work on a whale, a blue whale, as a model organism? Say you want to establish some facts in mammals, and you wanted to work on a dolphin, and you wanted to work on a whale, or you wanted to work on an elephant. Now this will be really difficult to tame an elephant, or tame a blue whale, or tame a dolphin, and keep it in captivity. You see, there are two simple concepts that we all know. One is in vivo and another is in vitro. So you can work in laboratory, but whether the biological system have similar set of observations and similar set of activities, you are not sure. So keeping an organism in captivity will be difficult if they are huge in size. Like I said, an abrupt example, an elephant, right? So small size sometimes makes it very easier for a researcher to carry out the experimentation. So one aspect will be size. Now, if you can recall Mendel's experiment, right? If you can unmute and somebody can tell me on what organism did Mendel work on with respect to.
Pisum sativum, pea
Absolutely,
plant.
absolutely right. The pea plant, Pisum sativum, garden pea. And there is that classical question which was asked in our class 12th examination. Why did Mendel choose pea plant?
Short
Anybody?
lifespan. Pl-
Absolutely.
Annual plant, short lifespan.
Absolutely, absolutely. Now you see, if Dr. Pingley is working on some organism, I'm already 44, and my life expectancy would be say around 70 years on an average. I have left with say around 25 years of my life. And if I go around, say, studying something in human beings from, say, embryological state to aging, I cannot wait for 70 odd years for a person to be studied, right? So it really becomes very difficult to work for with organism which have larger lifespan. And therefore, is it not possible that I work on an organism which has shorter lifespan and try to replicate the finding in animals which are having longer lifespan? The answer is yes. Therefore, shorter generation time is something that we tend to look in when we are choosing a model organism. Another important aspect, see, all of you, I do not know how many of you are on fellowships. Today, the government is giving handsome fellowships, but there were times when there were no fellowships, and even if there were, they were in the numbers of say around 2,000 rupees, 2,500 rupees or so. I worked on say around 9,000 rupees fellowship, but I understand that now these days you have fellowships in tune of say around 30,000, 40,000 rupees. That is a handsome amount. Imagine yourself without fellowships. And if you have to, culture these organisms, maintain these organisms all the time, even if you need to work on guinea pigs, the fishes, see, we carry out these practicals on fishes in our laboratory. They cost hundreds of rupees per kg. I mean, per kg, if you buy a fish, there are hardly two fishes which one can buy. And then students find it difficult to dissect them out because they're expensive, right? 200 and 300 rupees kg. Right. So similarly, in your say research duration of say 5 years, if you have to sacrifice around 50 fishes for that matter, it will cost you a lot, right? Unless and until you catch hold of some fisherman who's very friendly and is willing to give you one or two fishes every now and then, right? So another important aspect with these model organisms is that they should not be expensive. It should not bother your pocket much, right? Unless and until you are having very good grants. But these days, grants are also very difficult to get. I mean, your guide would be having some grants, then that is fine. Otherwise, difficult to carry out those research works, right? Then another important thing is imagine, that you're working on, say, some aspect, especially developmental biology, maybe some aspect of an organism, and the organism is laying one or two eggs, one or two offsprings, and while you were working, a part of that offspring gets damaged, or maybe the egg gets ruptured, or the egg is spoiled. What will you do? You will undergo frustrations कि यार एक और दो तो अंडे आते थे, उसमें से भी एक फूट गया, एक खराब निकल गया, अब मैं क्या करूं? Several times the PhD students and the researchers suffer a lot because the model animal or the animal or the organism they had chosen, they are either not able to culture them in the laboratory conditions in the captivity, or their multiplication rate is very less, or the number of offsprings which are coming out are very less. So another important aspect, as I said, is the number of offsprings that are produced. If they're high, it is good. Then, if you are specifically looking into the genetic and genetics related aspect, it is always advisable that the model organism shows a huge variety of mutants. And therefore, when I was asked to deliver this topic for you people, I decided that I will emphasize a little more on Drosophila just because that it has a lot of mutants available, right? Natural as well as induced mutants, right? Now, then there are certain model organisms where observations become very, very easy. like Drosophila again, or maybe zebrafish for that matter, almost a transparent fish. And Drosophila, I mean, you can simply put it under a dissecting microscope or stereo microscope, identify the mutants also, the males and females, look into the several other developmental aspects, for example, ectopic eye, etc. in Drosophila as well. So observation is very easy. I mean, you can just do it through naked eyes. No need to go for electron microscopy and similar other aspects all the time, phase contrast microscopy also. So without doing that, you can observe these animals, so it becomes very, very easy. Another very important aspect these days is genome sequence. Now, when you are studying these animals, you are interested if the data related to these animals, specifically with the aspect of genome and sequence, whether it is available or not. So several of the model organisms, as we will see, have their complete genome sequenced now. Ever since in, say, late 1970s, sequencing of the genome started, several of the organisms, many of which are the model organisms, established model organisms now, have got their genome sequenced. So the information with respect to the genome and the sequences is rapidly and quickly available with complete authenticity for you. So that is another added advantage. Now, many of these organisms, with the exception of, say, mustard plant, which has separate set of advantages in botany that we'll be talking about, share a lot of genes in common, specifically which are related to diseases in human being, for example, neurological disorders such as Alzheimer's disease and Parkinson's disease. You know, these are some neurological disorders. So several of the genes which are causing certain genes in human beings and the functioning of some other genes in these model organisms have been found to be similar. I'll give you a few interesting examples also as we go ahead with this presentation. Transformation is another important thing that one can expect, right from E. coli to, for example, Arabidopsis also. Transformation, they should be able to undergo transformation. That makes the studies more even in C. elegans that can be seen. A comprehensive online database should also be available if you want to consider a particular model organism. And a lot of new tools, for example, CRISPR, then next generation sequencing, they are coming up. So the studies on the model organisms from this aspect have also been carried out. And there are several of these organisms where there are either no or very few ethical concerns. So nobody is asking you, why are you carrying out a work on Drosophila, maybe a fruit fly, so you're not affecting any biodiversity as such, or maybe C. elegans for that matter, or yeast for that matter, right? So the ethical concerns are on several occasions are not there with a lot of model organisms. So these are the aspects, not all of them, but several of them make an organism a model organism, right? So what are the some of the established model organisms which have been studied, and what are those research areas where these organisms have been utilized? So looking at different kingdoms or the different groups of organisms, you would see that Bacillus subtilis is something that comes a lot in literature along with E. coli, right? So the chromosomal replication, especially of bacterial origin and cell differentiation, has been studied in Bacillus subtilis. Then in E. coli, conjugation and phage genetics has been studied. Similarly, if you come down to Neurospora crassa, if you remember, Beadle and Totem proposed this one gene, one enzyme hypothesis, which was later called as one gene, one polypeptide hypothesis. That was through the study of Beadle and Totem on Neurospora crassa, also, which is one of the famous fungal species, where meiosis, metabolic regulation, and circadian rhythm have been studied a lot. And another very, very significant, like I have already mentioned, is Saccharomyces cerevisiae, the baker's yeast, which we commonly call as the baker's yeast. Now, a lot of study has been carried out with respect to, if you can remember, CDK2 kinase, right? Cyclin dependent kinase, if you can remember that, the checkpoints in the cell cycle, all these studies have been carried out in Escherichia, and cell signaling studies have also been carried out. There's this algae which is called as chlamydomonas, where mitochondrial protein studies have been carried out. Then in plants, as somebody said, Arabidopsis thaliana is the first plant where complete genome sequencing was carried out and is famous for plant molecular genetics, apart from its role as, I would say, a model organism, model plant with respect to plant hormones also. We would see that further as well. Oryza sativa, the rice, which is known for a lot of work associated with genetic modification to increase the resistance and the production values in the rice varieties. Then we have already talked about Z maize, that is maize, where if you can remember Baba McClintock, transposons, developmental physiology, epigenetics, pest resistance, hybrid vigor, that is heterosis, quantitative inheritance, and comparative genomics. So Z maize has also seen a lot of work that has been carried out with respect to the plant kingdom. Then in animal kingdom, there are again several animals, amongst which the four which are mentioned here are a lot famous and favorites amongst the researcher. C. elegans is known for the studies on the genetic control of development and physiology. Then zebrafish, Danio rerio, is associated with the developmental and evolutionary study, famous for evo-devo concept and specific gene function. And we'll be talking a bit more on Drosophila today. So, like I said, Morgan's study, if you could remember, the sex-linked inheritance, if you could remember, genetics, physiology, microbial pathogenesis, life history, evolution, all the aspects. In fact, a lot of I have some graphics to show you today where you would see that a lot of biological aspects in human beings are almost copied in same way in Drosophila, right? And vice versa. And then the frogs, Xenopus laevis, where molecular developmental and cell biological studies have been carried out. So these are some of the organisms you should remember as model organism, right from prokaryotes to eukaryotes and vertebrates and mammals, right? Going further. Now, since I said we are talking about the model organisms, and one of the aspect of model organisms was um, the life cycle. We were talking about short lifespan, right? So see how lifespan in some of the model organisms differ. Cell cultures, we are not talking about the lifespan because it is in vitro thing that you are doing. Now, if you look at this lifespan of C. elegans, it is almost 3.5 days alone. Life cycle of Drosophila is around 12 days. Life cycle of a mouse is around 50 to 60 days. And life cycle of a zebrafish is around 70 to 84 days. So imagine your PhD or doctoral work. Imagine you are doing an MSc dissertation and you have 6 months of work. And if you are doing a doctoral work, you have to do work in replications. So several experiments in triplicates maybe. You're happy with C. elegans maybe. 3.5 days, एक बार life cycle खत्म हो गए, or you can start another set of experiment. 3.5 days, next point 3.5 days, another set of experiment. Fail भी हो गया तो इतना दुख नहीं होता है कि यार fail हो गया, चलो एक बार फिर से experiment करना है. But imagine कि किसी ऑर्गेनिज्म में 1 साल का लाइफ स्पैन है या 6 महीने का लाइफ स्पैन है, और 6 महीने काम करने के बाद आपको पता चले कि आपका एक्सपेरिमेंट फेल हो गया है. तो one aspect is that you had to wait for 6 months, and second aspect is after 6 months, if you come to know that your experiment has not worked out, you undergo a frustration and you start it afresh, right? So how many failures can you actually bear? Not many, right? But with a life cycle span of say around 3.5 days or 12 days, 50, 60 days odd, you're happy. Even if you fail in one of the experiments, probably you'll bother to repeat it again, right? You will not bother to repeat it again. So that is where the time to obtain the fertile adults again, that becomes handy, right? So this is one aspect that I wanted to focus on. Then another aspect is the brood size. Brood size. Here I have not focused on Arabidopsis. I'll be focusing in some different way later on when I'll be talking about Arabidopsis. Right. So brood size. When you are looking into an organism from the aspect of a model organism, you want high fecundity, right? You want to have that organism to be a fecund one, a larger brood size, because a research, a scientific finding becomes significant when you do it at a large sample size, right? Say for example, imagine that I'm working on Drosophila. I'm studying effect of something, some chemical, some heavy metal, something, some nanoparticle on the fecundity of Drosophila, right? Or maybe some generations of Drosophila, something, right? So if I have, say, one or two or five or 10 eggs, statistically, it does not remain significant for me to work on few eggs. So I'll be looking for more generations, and if I look for more generation, it will take more time. So I have one risk that either I don't have significant observations, and second risk is probably I will require more time. But like you can see here in C. elegans to zebrafish, there are 200 eggs per week from zebrafish, right? A lot of work goes in my laboratory under Dr. Borgave in the areas related to zebrafish. So breeding of zebrafish takes place. They're happy to get 200 eggs in a week. In Drosophila, I get 120 eggs a day, and same goes with C. elegans. You have 140 eggs a day. So you have a repeated next generation. So you could quickly get F1 generation, F2 generation, as many generation as you want. And then you have ample numbers in terms of sample size to make your observations significant. And if you compare that with mus musculus, you have 6 to 12 pups per month. Again, when we look into the mus musculus, that is mouth with the aspect of mammals, I would say it is still a high number, because comparing this with the gestation of 2 years of an elephant or 9 months of a human being, 6 to 12 pups per month is still a good number, right? So if you want a mammalian model organism, mouse is still a good choice. And zebrafish can still be a better choice with respect to the vertebrate biology, right? And as you go into the invertebrates, you have Drosophila and C. elegans for that matter, right? Now, this for me and for most biologists would be an important aspect. When you are looking at an organism from the aspect of model organism, especially in case of animals, you are interested to look into the homology of those genes which cause human disease, like I was addressing Parkinson's and Alzheimer's for that matter, right? So in case of cell cultures, the lines, there is 90% and more gene homology. But if you're talking about animals, then you have C. elegans with 65%, Drosophila with 77%, mouse with more than 90%, and zebrafish with around 84% gene homology for human disease. Please do not get mistaken that this is a genome homology or gene homology with human beings. It is gene homology only for human diseases. When we are talking about model organisms, then human diseases make an important aspect, an important area, because when we choose model organisms, we choose them mostly when we are interested to find the homologies with respect to the occurrence of the diseases in human beings also. A lot of work is already been carried out from that aspect. And this was another thing that I was talking about. See, because they play a pivotal role, or they hold a pivotal position in the evolutionary tree, the model organisms find a correlation. Now, the numbers which have been written here, 90, 310, 360, 450, 990, they represent how many million years ago these have evolved, right? So if you compare human beings and C. elegans, you would see that C. elegans Drosophila, for that matter, are quite distant to human beings and mouse from, say, around 900 million years ago, right? So you would say that C. elegans evolved 900 million years ago as compared to human beings and the mouse. And as you go closer, zebrafish, xenopus, and chicken and mouse, they find much more evolutionary resemblance to the human beings, right? So that aspect is also studied when you're looking into a model organism, right? So as you go further, then the resemblance gets lesser, and you might not see the same homology or the rate of homology in these organisms also. Another aspect that I was addressing while talking about the model organism was the sequencing of the genome, complete sequencing of the genome. So I'm presenting here the timeline. Now, you see, I said that it was in the late 1970s when the genome sequencing. Now, genome sequencing has also seen a lot of revolution. The present phase that we see is the next generation sequencing, that is metagenomics, and omics is also coming in, right? So when 1920s it started, bacteriophages ms2 and 5x174 were the ones which were sequenced first. Can somebody tell me, unmute and tell me, why do you think, was it a matter of chance, or was it something else that these were sequenced first? What do you think would be the logic behind this?
Lesser number of chromosomes.
Yeah, there's a number of chromosomes, or small genome size. Correct. The genome was small, so one could sequence the small genome. That was the reason that bacteriophage. It all started with bacteriophage, and gradually it took around next 20 years for us to sequence bacteria. So Haemophilus influenza, Methanococcus janashi were the bacteria where the sequencing was carried out, and the next one was Saccharomyces cerevisiae, for which the genome was sequenced in 1996. Now here onwards, keep them apart, the bacteriophage and the two bacteria which were sequenced. Now this onwards, Saccharomyces cerevisiae, that is the yeast, Baker's yeast, 1996, then Caenorhabditis elegans 1998, then Arabidopsis and Drosophila 2000, Homo sapiens, human beings, you all know Human Genome Project, so 2001, Mus musculus, and Takifugu is a pufferfish, Japanese pufferfish, so 2002 that came up. Then 2004, Gallus gallus, the hen. Then Oryza sativa and Pan. Pan is a chimpanzee, 2005. Xenopus is a frog, 2010. And Danio is the zebrafish, right? So if you look at these names, you would see that after Methanococcus, Saccharomyces is a model organism. Caenorhabditis is a model organism. Arabidopsis is a model plant. Drosophila, model organism. Mus musculus, model organism. Oryza sativa, model organism. Xenopus, model organism. Denio, model organism, right? So several of these for which the genome has been sequenced have established them either before the sequencing of the complete genome, or after that as the model organism. So the simplest reason is that you have a whole lot of information with regards to these organisms and their genome available. The database is available, so you can explore it and correlate it with human beings. The human genome is already there, right? So this is another important aspect that I wanted to focus on. Now we'll start with the first one. I deliberately skipped the prokaryotes.